my channel Sam Biology Class. Friends, today we will discuss about gene cloning. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Gene cloning is a process by which identical copies of a specific gene or a DNA fragment is produced by using genetic engineering techniques. There are two different methods of gene cloning. First one is in vitro gene cloning. It is also known as cell-free gene cloning, in which the gene is amplified by polymerase chain reaction without involving cells. Second one is in vivo gene cloning. In this technique, living organisms such as bacteria, commonly E. coli, is used to replicate the gene of interest. This technique is also known as cell-based gene cloning. Cell-based gene cloning. Today we will know more about in vivo gene cloning. These are the key steps. First step is isolation of the gene of interest. Second step, vector selection. Third step, creating recombinant DNA. And fourth step, post-cell transformation. And the last step, selection and amplification. For gene cloning, the first step is to isolate the gene of interest. For this, a suitable tissue sample is taken from which the genome is extracted. This is the genome. Following this, the genome is cleaved by using suitable restriction enzyme. Restriction enzymes are endonucleases. They are the molecular scissors that cleave DNA at specific recognition sites. So cleavage of the genome by restriction endonuclease results in the formation of a number of double-stranded DNA fragments from which we have to isolate the particular fragment that contains the target gene. In next step, the gene will be inserted into a suitable cloning vector so that it will be able to replicate in bacterial cells. Next step is vector selection. As already mentioned, in vivo gene cloning, living organisms such as bacterial cells are used to replicate the target gene. But when foreign DNA is injected into bacterial cells, these fragments fail to replicate. This is due to the fact that these fragments lack specific sequences which are essential for its maintenance and replication in bacterial cells. Moreover, these fragments are degraded by different restriction enzymes that are naturally found in many bacteria. This problem is overcome by using suitable cloning vectors. Cloning vectors are DNA molecules that are used to carry the DNA into the bacterial cell where it replicates along with the gene. Different cloning vectors are available for this purpose. This includes plasmids, lambda-phage, yeast artificial chromosome and bacterial artificial chromosome. One of the widely used cloning vector is a plasmid known as PBR322. It is a circular double-stranded DNA molecule. It contains two antibiotic-resistant genes that are used as marker for the selection of transformed cells. Out of these two, one gene is for ampicillin resistance, one for ampicillin resistance, and the other gene is for tetracycline resistance. In addition to this, PBR322 contains one origin of replication and multiple restriction sites at which this plasmid can be cleaved by using restriction enzymes. After vector selection, the next step is to create recombinant vector or recombinant DNA. For this, the DNA fragment containing the target gene is inserted into the cloning vector. The ampicillin resistant gene of PBR322 contains a recognition site for the restriction enzyme PST1. So when treated with PST1, cleavage takes place within the ampicillin resistant gene. As a result, the circular plasmid is opened and become linearized. After cleavage with PST1, the particular DNA fragments containing the specific gene is inserted into this linearized plasmid. When DNA fragments are mixed with a sample containing PBR322, restriction enzyme PST1 and the enzyme DNA ligase, the DNA fragments will be inserted into some of the linearized plasmid, thereby forming the recombinant vector. The vector which contains the DNA from other source, that is the target gene, is known as the recombinant vector. At the same time, some of the linearized PBR322 are recircularized, thereby forming the non-recombinant or the normal vector. So after mixing, we are getting two types of vectors. One is the recombinant vector and the other is non-recombinant vector. In recombinant vector, due to the insertion of the target gene within the gene for ampicillin resistance, the structure and function of this gene is disrupted. This process is known as insertional inactivation. On the other hand, in the non-recombinant or the normal vector, both the antibiotic resistant genes remains intact. In this process, the enzyme DNA ligase, which is also included in this mixture, will join the ends of the linearized plasmid with the ends of the target gene. Next step is host cell transformation. In our example, E. coli is the host. In place of E. coli, other bacterial cells or even eukaryotic cells, if found suitable, may also be used. For host cell transformation, this mixture containing both recombinant and non-recombinant vector is mixed with E. coli and incubated. During incubation, transformation takes place, as a result of which some of the E. coli cells get transformed with non-recombinant vector, some with recombinant vector, and other E. coli cells remain non-transformed. After transformation, next step is selection and amplification. For this, we have to identify the bacterial cells that are transformed with the recombinant vectors. This is because the recombinant vector contains the cloned gene. First of all, we have to kill the non-transformed cells. For this, This mixture containing different types of E. coli cells are plated on a nutrient medium containing antibiotic tetracycline and incubated. As the non-transformed cells are sensitive to tetracycline, they will get killed. Remaining two types of transformed cells, one with non-recombinant vector and other with recombinant vector, will survive and will be able to multiply, thereby forming two different colonies. Out of these colonies, one type of colonies are formed by E. coli cells with recombinant vector, and remaining colonies are formed with E. coli cells with non-recombinant vector. In order to identify the bacterial colonies that are formed by E. coli cells with recombinant vector, these bacterial cells are plated on a secondary plate containing the antibiotic ampicillin. These bacterial cells are transferred to the secondary plate by using a nitrocellulose membrane. After transfer, when the secondary plate is incubated, numerous colonies develop. But these are the colonies that are formed by E. coli cells which are transformed with non-recombinant vector. Because the nutrient medium contains ampicillin, so the bacterial cells that contains the recombinant vector gets killed, so they fail to form colonies. By comparing this master plate with replica plate, we can easily identify as to which colonies contain the bacterial cells that are transformed with recombinant vector. After identification, these colonies are removed and then cultured in a fresh nutrient medium for a suitable time period. As a result of this culture, we will get a very large number of E. coli cells, each of which has the recombinant vector. Each recombinant vector contains the clone gene. So all the cells that are transformed with recombinant vectors constitute a clone. This process is known as gene cloning. Applications. Gene cloning has several applications. Number one, it allows for the production of a large quantity of industrial and therapeutic proteins such as enzymes, hormones, monoclonal antibodies, cytokines, blood clotting factors and others. Number two, this provides the specific gene for creating transgenic organisms. And finally, gene cloning enables the study of structure, function and regulation of specific genes. Friends, this is all about gene cloning. For more updates, subscribe my YouTube channel, Sam Biology Class.